Time course infections of HeLa cells using Vaccinia virus begin by setting up the viral infections, infecting the cells, and then harvesting the RNA from the infected cells at several time points post-infection. This RNA can later be amplified for hybridization to microarrays for analysis of gene expression.
Hi, I'm Judy and from the laboratory of Kate Rubins at the Whitehead Institute for Biomedical Research. Today we will show you a procedure for a time course infection with the Vaccinia virus in mammalian cells and the subsequent RNA extraction from the infected cells at different time points post-infection. This technique is a beginning step in a larger protocol aimed to extract and amplify RNA in order to assess virus gene expression during infection, which we will complete in 2 other JoVE videos. So let's get started.
To set up for the infection, grow HeLa cells in flasks and wait until the cells are approximately 80% confluent. Next, be sure to prepare enough viral growth medium for the experiment using regular DMEM with 2% FBS and no added antibiotics. The infection can be performed with either a crude stock of Vaccinia virus with a known titer or sucrose purified virus with a known titer if you're concerned about host gene expression. If you are using sucrose purified virus, skip to the next session. If you are using a crude Vaccinia virus stock, sonicate an aliquot of the virus just before infection in a cup sonicator with a bath of mostly ice and a little water. Sonicate it around 30 W for 20 seconds. Vortex the tube and repeat the sonication 3 times, adding more ice if needed.